we are in focus.
Okay, we're good.
We're good to go? Psych. So it might be hard to hear me, but we're going to review for test number 1. Review. Okay? Here we go. Now listen, honest to God, if all of a sudden you can't see this, you're going to have to say something.
Can't see it on the screen.
You can't see it now? Why? Because it's too small?
Right.
All right, well, you could see it before though.
Yep. That's good.
Has to be big.
Yep.
Big. And less bright, do you think?
I don't think that's helping.
All right, well, I'll just do that. All right, so how much paper can we go through? Number 1. So you know what? It's going to be a delay between when I have my hand there, I think, right? So number 1 is the definition of homeostasis. So I'm not going to write everything down, but homeostasis is to maintain a constant internal environment, right? Homeostasis, maintain a constant internal environment. The number 1 way that we maintain homeostasis is via negative feedback, right? In negative feedback, the change in the variable is in the opposite direction. That's very important. So my example, if your blood pressure is going up because you have anxiety, because the pen doesn't work on the computer console, via negative feedback mechanisms, your blood pressure will go back down, hopefully to control levels, to normal levels. But the change is in the opposite direction. That's very important. Occasionally, homeostasis is maintained by positive feedback, and in positive feedback, the change is in the same direction. The same direction. And what were our examples of this? Childbirth and a little bit of blood clotting. Childbirth and blood clotting. So there aren't very many examples of this. But remember, with positive feedback, that change will be in the same direction. It makes things worse. It exaggerates the situation. If you have any questions, you're going to have to yell them out, because I can only see 3 people. All right, so moving on, number 4 here we have serous membranes. And in serous membranes, I want you to remember that the liquid in a serous membrane is water-based. Okay? It's usually acting as a lubricant. In addition, when we talk about serous membranes, I can't call up a picture, but I gave you the example. The best example is that picture in your book where they take a fist and push a fist into the balloon. So with that example, oh my gosh, here's your fist. That's so bad. Here's the balloon though, okay? It's bad, it's busted. So the part of the balloon that touches your fist, that part is the visceral layer. That's the inner layer. And then the outer layer is the parietal layer. Uh-oh. I don't know. That is totally spelled wrong. Parietal layer is the outer layer. And then when it comes to serous membranes, you should know pericardial. That has to do with the heart, right? Pleural are the lungs. I'm not going to try to draw lungs. And peritoneal means digestive organs. So 3 places that we find the serous membranes: the pericardial sac, you have a visceral pericardium, a parietal pericardium, you have a visceral pleura, parietal pleura, a visceral peritoneum, and parietal peritoneum. Different locations. You should know the locations. Okay. Any questions on that?
It says identify individual cavities.
It does? Where does it say that?
I don't know.
Oh,
I might have it.
yeah. Okay, that's a different. That's a different thing. I think that's the dorsal body cavity. I don't know why it says A, because there's no cavities here.
Yeah, that's what I was wondering.
Yeah, but I'm glad. Good, Rebecca. But for this Serious, are we good?
Yes.
Thank you. So as far as the various cavities go, we have two cavities. We have the dorsal body cavity, two major cavities, and the ventral body cavity. Remember, the dorsal body cavity is posterior, and it has two divisions, cranial, which houses the brain, and vertebral, or it has the spinal cord. And the whole entire dorsal body cavity is enclosed in bone. Enclosed in bone for protection. All right, the ventral body cavity, which is anterior, has two or three subdivisions. It has the thoracic body cavity, which houses the heart and lungs. Okay. Then there's the diaphragm that separates that from the abdominal. And then you have the pelvic. Now remember that the abdominal and pelvic body cavities do not have a separation between them, so sometimes they're called abdominal pelvic.
Is the diaphragm technically the thoracic or the abdominal?
That's a tough question. I don't know the answer to that. I think it depends what you read, which one they put it in. I would probably put it in thoracic, but everything you read always says it's a separation between the two. But I would put it in thoracic if I had to choose. Kyle.
Got it right this time.
Yeah, you're right this time. Okay. Actually, since we're here and talking about the body cavities, make sure that you remember that the abdominal pelvic cavity can be divided into 4 quadrants, right? So you have upper right, lower right, upper left, lower left, and then you should have an idea of 2 organs or structures in each one. You cannot include small or large intestine or the word colon. Remember that? That's on your lab quiz today, actually, too. And you would not be using lungs or heart or trachea or something like that, because that would be in the thoracic body cavity. All right, so this thing here, I wrote basic anatomy.
This dude's got a question, he's just too shy to say anything.
Okay.
What would be an organ in the lower left?
Lower left would be, you could say bladder.
Okay, I wasn't sure if you said
Seven.
bladder.
Yes, you can. It's in both.
Okay.
Bladder, seminal vesicles, left and right. You
Okay.
could say fallopian tubes, uterus, ovaries, ureters, kidneys, yeah. These are kind of both, so yeah. So there's a lot that you can say. And that there's a mirror image. In the lower right, though, you could add cecum and appendix. That's what makes that unique. On a stiff neck by the end. All right, and basic anatomy is kind of like just what I just said. You need to have a clue where the heart and lungs are, where the abdominal organs are, just a clue where these parts are. But also when we're talking about basic anatomy, remember I mentioned the difference between arm and forearm. And then the difference between thigh and leg. Okay? So that's, in my book, that's basic anatomy, that you would know that. Has a video. Thank you. All right, so then we're on orientation, directional terms. So you want to make sure you can use words like superior, inferior, anterior, posterior, medial, lateral, superficial, deep. Any questions on how to use any of these words? Also, I want to add to that proximal and distal. Those are, I think, the hardest. You can only use these for your limbs. Proximal is closer to attaching to the body, and distal is further away. Further away from being attached. Anybody have a question on that? Make sure you can use those words. This is just a review, so I'm not going to go over every single thing. Oh, also regional terms, like there's two type, regions to our body. The axial region, which is the head, neck, and trunk. And then the appendicular, which are the limbs and their attachments. And then the last part for this chapter would be to go over the body planes. I hope that after this I hit everything. I think so. I don't know what else there was. Remember, you can cut the chick into those 3 different planes. The easiest, I don't know, like the fact that I could even, right? So the easiest is the transverse. That's cutting someone transverse into a superior and inferior plane, right? That's like decapitation, cutting you off at the knee, cutting you in the horizontal plane, superior and inferior halves. Then you have sagittal. In sagittal, you're cutting someone into a left and right half. Left and right half. And if it's equal left and right halves, it's called mid-sagittal. And if it's off-center, it's called para-sagittal. And then finally, it'll be very hard for me to draw this, but if you cut someone, I can't do it. It's not even right to draw that. Then the next one is, it's wrong. Frontal, or they also call it coronal. Cutting you in the frontal or coronal plane cuts you into an anterior and posterior half, where your face is on one plane and your butt is on the second plane. Okay, anterior and posterior halves. It's called frontal or coronal. All right, so that's a quick little review of chapter 1. You know, please also read over the notes just in case there's something I missed, but that sounds almost like everything, right?
For hierarchy of organization.
Oh, there's no hierarchy. I missed it. Thank you, Rebecca. So important. That's a question for sure. That's on the final exam. Hierarchy of organization. So that means you start with an atom, go to a molecule, and you have organelles, which we talked about today. Then you have a cell, which is the structural and functional unit of life. Then a couple of cells together forming the same function as a tissue. There's 4 tissues: epithelial, connective, muscle, and nerve. Then you have an organ, which are 2 or more tissue types working together. Then an organ system. And then finally the whole organism. Which is you. Thank you for pointing that out. So the kind of question is either put in the right order or which is larger, which is smaller out of this list. Anybody, anything? Can't say anyone. Bueller? Anyone? Okay. All right, so chapter 2, we are doing our chemistry. Basic chemistry. So remember, like I always say, this is a review. It doesn't take place after the whole lecture. We're just going over the various parts of an element and of an atom. So remember, atoms are the smallest part of an element that retains the properties of the element. It would be the atom, the smallest part of an element that retains the properties of that element. And then we have the subatomic particles, a proton, which is a positive charge, a neutron, which has no charge, and an electron, which is negatively charged. And if your atom is neutral, then the number of protons will equal equal the number of electrons. It's the number of protons that determines which type of element we have. So if you, let's just say if you have 11 protons, for sure you're going to be sodium. If you're neutral and you're sodium, you also have 11 electrons. The number of neutrons can vary. That actually is what makes up isotopes, having different numbers of neutrons. All right, also remember that, in a quick example, that the protons and the neutrons are located in the center of the atom, the nucleus, and then traveling around in these shells or orbitals are the electrons. And really what electrons want to do, if you're going to give them any humanistic qualities, is to have their outer shell to be filled. When the outer shell is filled with electrons, it is stable and tends not to make bonds. And then remember, it's the electrons in the outermost shell that will determine what type of bonds and how many bonds one atom will make with another atom, right? You guys remember that part? It's the outermost electrons. These outermost electrons, by the way, are called valence electrons. The valence electrons. So the electrons in the outermost shell, and they determine what type of bonds are made. Had I known I could be in this little desk area, I could have been happier. But then I couldn't show you my pictures. But it's kind of cozy. Down here. So 3 major types of bonds, ionic bonds. In ionic bonds, there is a complete transfer of electrons from one atom to another, a complete transfer. And so when you're done, you make ions. Ions are charged atoms. And there's 2 types of ions. You can be a cation ion, which is positively charged, it has that positive in there, or an anion, which is negatively charged. Remember, to become an ion with this complete transfer of electrons, there's only going to be the movement of electrons, no movement of protons. So to become a cation, you had to lose like that rather, electrons. And to become an anion, you had to gain electrons. Okay? I don't know what else to say about that. That's it, right? Best example is salt, sodium chloride. In covalent bonding, that's where your electrons are shared. And there's 2 ways to share. The nice way, and I'm joking, non-polar. It means there's equal sharing of electrons. While polar is unequal sharing. Or the best example for equal sharing would be lipids. Things that are hydrophobic. Water, insoluble. While equal sharing, unequal sharing would be water itself. Things that are hydrophilic, water soluble. How's it going? Can you read my handwriting all right? It's being recorded, FYI. Hopefully. It's recording, right? Though it's like red recording.
It is, yeah.
I don't trust anyone. I have to have triple checks because I had so many pictures of people just swearing to me that was not the picture. So. Don't believe people that lie about your head. And then the last would be hydrogen bonding. In hydrogen bonding, we see that between a hydrogen that's in a polar covalent bond and another atom in another polar covalent bond. And our best example would be between water molecules, between water molecules. So if you drew water like this, and you draw it like this, remember, and not straight lines, but a little angled, because water has a lot of two polar covalent bonds in it, unequal sharing. So you kind of draw it like that. So there's this one water molecule, and then here's the second water molecule. And then it's the attraction between the hydrogen and the oxygen. That is a hydrogen bond. It's a very weak bond, easily formed, easily broken. So you have two questions here. Bond number 1 and bond number 2. Bond number 1 is within a water molecule. That is a polar covalent bond. Bond number 2 would be between two water molecules. That is a hydrogen bond. Okay? You don't want to get those two very easy questions wrong. Water leads us to talk about pH. Remember, pH is a measurement of the free hydrogen ions. It measures the free hydrogen ion concentration in a solution. It's got a scale of 0 to 14 with no units. If the pH is less than 7, your solution is considered acidic. If your pH is equal to 7, you are considered neutral. And if your pH is greater than 7, you are basic. Okay? And there's an inverse relationship between hydrogen ion concentration and pH. As you increase the concentration of free hydrogen, you will lower the pH. So something that has a pH of 2 has more free hydrogen than something that has a pH of 10 has less free hydrogen, because of the inverse relationship. Make sure you also know the definition of an acid and a base. Acids add free hydrogen to solutions. Bases remove free hydrogens. And a buffer. Buffers try to maintain the pH, keep the pH constant. Buffers try to keep the pH constant. Acids add free hydrogen, bases remove free hydrogen. All right, exergonic and endergonic reactions. So endergonic reactions. These reactions you have to add energy, energy. That's funny. Exergonic, so ridiculous. It's my own little world. Exergonic reactions. Release energy, NRG, energy. The abbreviation. Funny, haha. Working on my own up here. Not on myself. And then we also, since we're here, we also had synthesis reactions. Might as well talk about that. Synthesis and decomposition. Synthesis are building reactions. They're anabolic. Building, making something bigger. And decomposition would be catabolic, breaking something down. And the synthesis reaction we talked about is dehydration synthesis. And the decomposition was hydrolysis. Rebecca, does anyone have any questions? Thank you, Rebecca. All right, so on our organic molecules, we have carbohydrates. You have to know the building blocks for every one of these. So carbs, the building blocks are called monosaccharides. 2 of them together are a disaccharide, 3 of them together a trisaccharide, and then polysaccharide would be many together. And under polysaccharides, you should know glycogen. That's how animals store carbohydrates. Starch is how plants store carbohydrates. And cellulose, plants use that. We cannot break it down. For us, that's fiber. Carbohydrates overall are very polar, hydrophilic, water-loving. And they're used for quick energy, Justin. Lipids, a little bit more complicated. There are 3 types of lipids. You have the triglycerides. There's one that's made up by 1 glycerol and 3 fatty acids. Make sure you realize that the fatty acids can be saturated with only single bonds between the carbons, or unsaturated, where you have at least 1 double bond. And triglycerides are usually a way that we store energy for long term. Triglycerides. Phospholipids, another type of lipid. They're made up of 1 glycerol, 2 fatty acids, and a phosphate group. What's important about phospholipids is, because triglycerides, by the way, are very nonpolar. And in general, all lipids are nonpolar, except phospholipids, which are both nonpolar and polar. These are the phosphate. So that means they are amphipathic. That means they're polar and nonpolar. They have both. You find these in the cell membrane.
What does that say?
That says amphipathic. I don't know. There's an H there. I spelled that really wrong. But
What does that mean? That this one is polar?
it's polar and nonpolar. It's hydrophilic and hydrophobic. It's got both polar and nonpolar parts.
Can't see the list.
No, it's good.
Yeah.
It's just like being in the class, man. You watch that video. Get to hear people opening up their sandwiches and everything. It's nice. All right, proteins. Building blocks of proteins. Oh wait, steroids. What?
I was gonna say, isn't there a third?
There is a third one. Steroids. Thank you very much. Steroids are a 4-ring structure. I can try to draw them, but I have no idea what I'm drawing. Why? Why am I doing that? 4-ring structure, and then they have different things stuck on here, like an oxygen or something. I don't know, whatever. They have different things. So the precursor to all steroids is cholesterol, so you need to know that cholesterol is a steroid, and everything comes from cholesterol. And then remember that certain hormones are steroids, like the sex hormones, and other hormones like cortisol, aldosterone. They're a type of lipid called a steroid. Bile salts. All right, back to proteins. The building blocks of proteins are called amino acids, which there are 20 naturally occurring amino acids. Amino acids vary from each other in the R, or variable group, that's what it's called. The R, or variable group, and then there's 20 different things that can be added to a given amino acid to make 20 different amino acids. There is a primary, secondary, tertiary, and quaternary structure for proteins. The primary structure is to bind one amino acid to the second amino acid, to the third amino acid, to the fourth amino acid, and so on. And the name of the bond between each of these is a peptide bond. It is a type of covalent bond. It's a strong bond for the primary level of structure. The secondary level of structure, you have an alpha helix and a beta pleated sheet that's held together by hydrogen bonds, which would be weak bonds. The tertiary level of structure is usually globular, and it is a variety of bonds. You need to get to this level for the enzyme to be functional. You usually need that tertiary structure,